Hello students, in this class let us learn about the working of a travelling microscope, another length measuring device. So you can see the picture of a travelling microscope here. You can see this device in the lab. We use this device to measure lengths such as the diameter of a capillary tube or the shift in certain heights or the capillary rise and so on. Now let us understand the various parts of this travelling microscope. As the name indicates, this consists of a microscope which is able to travel both vertically and horizontally. So in this picture, this is the microscope piece, and these devices, these attached vertical section and the horizontal section enables this microscope to travel left to right and up and down. So for this microscope, we have an eyepiece and an objective, and the distance between this eyepiece and the objective can be adjusted using a rack and pinion arrangement. So that means you can adjust the focus of this microscope using this rack and pinion setup. And in order to measure the length, we need additional setups of scales. We have a vertical scale here, and when the microscope moves up and down, we normally measure the vertical scale readings. And here we have a horizontal scale here. When we move the travelling microscope left and right, we take the horizontal scale reading. And on the vernier scale itself, we have a main scale as well as a vernier scale. So you have to take the main scale reading as well as a vernier scale reading. Similarly, on the horizontal scale, we have the vernier scale and the main scale. So you have to take both the readings. At a time, we will be focusing on one particular scale depending on which motion we are applying to the microscope. Sometimes, instead of this vernier scale, we will have an arrangement of a screw gauge on this microscope. In that case, we will be using the normal procedure for a screw gauge. So as for now, let us consider the case when we have the vernier scale on the travelling microscope. So this is the image, real image of a travelling microscope. This is the microscope part. We have the eyepiece, objective, rack and pinion arrangement. Then there is the vertical setup. So you can see here, this is the main scale. Here this is the vernier scale. Here, in addition to that, we have the screw gauge setup where additional markings on the head scale are made. So here this is the main scale, horizontal main scale. Then there you can see the vernier scale here and so on. Now, how do we measure distance using travelling microscope? Suppose if we want to measure the height of this pin, when we hang a weight over here, this pin will be shifting down, right? This cantilever will be bending and this pin will go down. Suppose we want to measure the shift in the height of the pin when we hang a weight over here on the hanger. For that, we will focus the travelling microscope onto the tip of this pin. And while you look through the travelling microscope, in the eyepiece, we have a crosswire marking like this. And these crosswires can be made to coincide with the image of the tip of the pin that you see. So in this particular case, you will have to coincide the horizontal crosswire at the tip of the pin. So when you hang the weight onto the cantilever, this pin will be going down and the horizontal crosswire will remain at the same position. Then you move the travelling microscope to again coincide the tip of the pin with the horizontal crosswire. So you take two readings. In the first case, you take the reading here without the weight hanged, and after the weight hanged, you take the second reading, and the difference in the readings will yield you the shift in the height of the pin. In another case, like measuring the diameter of a capillary tube, so here you can see this is the cross section of a capillary tube, this is the horizontal crosswire, and this is the vertical crosswire. In order to measure the diameter in this dimension, First, you will coincide the horizontal crosswire at the bottom part of this cross section of the capillary tube. You take reading number 1, then you move this microscope up and enable the horizontal crosswire to coincide with the upper part. Then you take the reading 2. Reading 1 minus reading 2 will give you the diameter of this capillary tube. Similarly, you can do the same with the vertical crosswire. The vertical crosswire can be moved and made to coincide with the left side of this capillary tube. Suppose you take reading number 3 with that. Then you move this vertical crosswire and enable it to touch with the right side of this capillary tube, and you take reading number 4. 3 minus 4 is also the diameter of the tube, but in another dimension. Here you measure this diameter. In the first case, you measured diameter in this direction. In one case, you move the microscope up and down. Here, microscope is moved up and down to move the horizontal crosswire up and down. And in that case, you take the reading of vertical scale because you are moving up and down. Here you move left and right. The microscope moves left and right, and in this case, you have to take the reading of the horizontal scale because horizontal scale enables the motion of microscope in the left and right direction. So this is how you take readings on a travelling microscope. So we have discussed the various parts of a travelling microscope, and taking the exact readings on the scale are very similar to a normal vernier scale reading, just like a vernier calipers, or sometimes just like the screw gauge. Other than that, we saw how we measure actually take the readings by coinciding the crosswires. I hope this portion is clear for you. Thank you.